right. So welcome everyone to the first session of the Advanced Computational Medical Technology Seminar by the John Curtin School of Medical Research for the year 2022. Today I'm very pleased and very happy to have Reto Fiolka from University of Texas Southwestern Medical Center at Dallas. He's currently holding an assistant professorship in bioinformatics and cell biology. And Reto has been very prominent in the field of advanced imaging, and most recently done a lot of work in a modality called light sheet microscopy and definitely super resolution. But Reto sort of came from a very different beginning. He was a mechanical engineer by training in computational fluid dynamics. He completed his PhD in the Institute of Nanotechnology at ETH Zurich in the group of Dr Andreas Stemmer, and really moved into the field of optics at the Hughes Medical Institute at Janelia Research Campus, and working with a founder, in fact, in super resolution technology, SIM, Mats Gustafsson, working on 3D structured illumination microscopy, and then follow on working, I guess, in Janelia as well with Meng Cui on adaptive optics. And now he heads a group in UT Southwestern, where he extends a lot of his imaging capability to look into cancer research and looking at obviously models that are physiologically relevant, three-dimensional, ex vivo and vivo. So I think today he's going to speak a lot about his pet project on light sheet microscopy and how it plays a fantastic role in improving the spatial and temporal resolution for all bio imaging system at different size scale. So Reto, please take it away. Thank you.
Thank you, Steve, for the kind introduction, and thanks everyone for joining. Good morning. Well, here it's evening, but good morning in Australia. Yes, I will talk a little bit about some recent work we have done to improve both the spatial and the temporal resolution in light sheet microscopy. And here is an image of a cardiomyocyte that was imaged with a new light sheet technique that is combined with structural illumination, itself a super resolution technique that, as you heard, pioneered by my former advisor, the late Mats Gustafsson. So what will I talk about? I will spend some time talking about what is light sheet microscopy and what is structural illumination microscopy. Just a little bit of the theory in case some in the audience are not aware of it or may benefit from knowing a little more about it. And then I will jump in and show how we combined the two, which we called oblique plane structural illumination microscopy, a name that hopefully will make more sense when we get there. And in the second part, I will talk about the projection imaging technique that in principle can be added to any light sheet microscope that then allows faster acquisition rates when you are looking at a 3D sample. All right. So what is a light sheet fluorescence microscope? Here is kind of a 3D model of one. And these types of microscopes were invented by Ernst Stelzer back then at the EMBL in Heidelberg, when he was tasked to make, I think, a bit more practical microscope. He made very stunning microscopes, among them the 4-pi microscope he was involved. Then I think the idea was to build a microscope that can be really employed in many research fields. And if you look, what's the trick of the light sheet microscope? Somewhere here, this capillary is the sample. So if you zoom in, the idea is basically that with one objective, you create a sheet of light that illuminates this sample here in yellow, and it illuminates a slice of it, and that excited slice is then imaged with a second objective onto a scientific camera. In principle, very simple, or at least in that form. And here, if say the sample is three-dimensional, like the zebrafish embryo, the idea is that the light sheet just illuminates a section of it, and hence doesn't illuminate stuff above and below. And as we will see, that is actually greatly beneficial in terms of lowering the rates of photobleaching and phototoxicity. But in principle, it is not too complicated, or at least in the first embodiments, it was a rather simple technique and a very versatile technique, and it found numerous applications, as we will see. So why would you build such a microscope instead of, say, using a confocal microscope? So here we can think of that the fluorescence photons we can squeeze out of a sample are finite. And for many people, it makes more sense if we think of that as a budget, as kind of money you have on the bank. So think of your bank account and say you have about $10,000 on it. And then the analogy of taking a single image was kind of, you make a withdrawal of say $1 and the bank charges you $100 commission. The commission would essentially be the photons that you don't use, but nevertheless excite. So that would be a terrible bank, because if you were to make multiple withdrawals, which in the analogy would be like acquiring a Z-stack, you would have exhausted your entire bank account after withdrawing like 100 Z slices. Again, in this silly argument, each Z slice would be worth $1 and the commission would be $100. So obviously you wouldn't do business with such a bank, and that drives it kind of home what we're actually doing with, say, a confocal or wide field microscope, because the majority of the excitation is kind of in vain, is kind of lost. So the few photons you actually collect from your focal plane are offset by a lot of photons that you excite that you're then not using. And so the light sheet microscope is close to a bank that we're actually using. So when you make a withdrawal, you actually almost pay no commission. You only get photons that you actually use to form your image, and you don't withdraw any fluorescent photon from above and below. So you can essentially get them once you move the light sheet there and acquire the new image. And by the way, can you hear me? Do I need to speak up or actually calm down a bit?
It sounds good. Thanks.
Okay, it sounds all right. All right. So sorry for the very basic, or for most people it becomes much more clear when we talk about money. So sorry if I assume that. Maybe that's just the thing in the US, but nevertheless, when you say 90% of the fluorescence you excite in a sample in a confocal is not used, sometimes it doesn't drive home the point as much as if it's, kind of the money in your bank account that just goes away. Well, all right, enough of that. So this really works. This was early work with light sheet compared to a delta vision. So a wide field microscope imaging yeast, and this should be a movie. Here it goes. So these are stacks in the wide field. They bleach off very quickly. And here, sure, in the SPIM, which is another of many acronyms for light sheet fluorescent microscope, you can see you can image the yeast much longer. And also here quantified as kind of the intensity you get from it. So this actually works. Good. So there are hundreds of light sheet techniques, and one can argue, what's the common denominator, or what do they have in common? So one thing we can maybe spend a short amount of time on is how do they actually create the sheet? And I think there are at least 3 different variants that are worthwhile considering or mentioning, or you can argue you can construct all the other types from them. So the first one which is sometimes referred to as the SPIM, which stands for Selective Plane Illumination Microscopy, essentially shapes a laser beam into a one-dimensional focus. So at any time there is kind of a sheet, a static sheet there. Then there are the digitally scanned light sheets, which form a two-dimensional laser focus, or other beam geometries like bezel beams or optical lattices, if you want so. And they are being scanned, at least in one spatial direction, to form in the time average a sheet. So this was an acronym coined by Philip Keller, who pioneered this. So this is the DSLM. So in contrast to the SPIM, the DSLM kind of in the time average creates a sheet of light. And then lastly, you can also create a SPIM light sheet and actually scan it in the orthogonal direction to here. So essentially scan it in the direction it propagates, which would be here, I think the Y direction. But as you can see, here maybe in the cross section, the sheet moves here from left to right. And this is sometimes called the axially swept light sheet, or also the tiling light sheet. So I think these are kind of the three forms that are predominantly used to create light sheets. And they are, as I mentioned, they're by now over 100 different techniques. And I'm guilty as charged. We also coined a few acronyms in that field. So let's maybe leave that for the light sheet part and talk a little bit about what is structural illumination microscopy. So the second ingredient, or the second technique I want to talk about. So assume here on the left, these fringes were your sample, your sample structure, or more specifically, your fluorophore distribution. Just for a moment, think of these wavy lines as the sample, and also assume that the spacing of these fringes might be too fine to be seen. And here on the right, this would be the distribution of the excitation light in this form fine stripe pattern. So if we actually superimpose the two, then you can see an effect that is called moire fringing, that the beading of the frequencies in the sample and the single frequency here of the illumination together create a pattern that has down converted frequencies. So you may notice there are coarser structures that haven't been there. Those are not the sample structure. Those are essentially the multiplication of the illumination pattern with the sample, creating these moire fringes. So one can construct the argument that maybe now, given that some of these frequencies have been down converted and made more coarse, or the structures in real space made more coarse, you might be able to observe them with a microscope, and with the knowledge of the illumination pattern, if you know precisely what frequency, what pattern vector, and what phases it has, you may be able to reconstruct what the sample has been. This is, if you want so, at the heart, the idea of structural illumination microscopy. We can talk a little bit more about the theory, and without doing any math, it can still kind of be explained what happens in a structural illumination microscope. Assume here that this is a computer simulated sample, or more precisely, this were the distribution of fluorophores. Here, so some stripe patterns, the Siemens stars, some dots and lines. And if you Fourier transform that, so if you put in reciprocal space, then this is the sample's Fourier transform. You may not be able to ever see it as such, but this would be kind of the true Fourier transform. And you can see, probably owing to the Siemens star, there are these spokes that radiate out from the origin. So the further you're away from the origin, the higher the frequencies are, and the direction also encodes in real space in which direction these fine features are. A real microscope, however, doesn't see the true fluorophore distribution. It essentially sees a downsampled version, a blurry version, as shown below. And in frequency space, this corresponds to a low-pass filter. So within this blue circle, those are the observable frequencies or Fourier components of the sample, and they're a subset of the lower frequencies. The higher ones you will not see. That's just limited by diffraction. So this out here is, if you want, so the AB diffraction limit, and higher frequencies simply cannot pass. So I said this is the fluorophore distribution. So this is not necessarily what the sample emits. If we don't excite it, nothing comes out. If we uniformly excite it, all the fluorophores will shine at the same time. And so there is a little loophole. So what if the illumination is a sinusoidal pattern with a fine fringe spacing, and here a zoom in, so you can see it has nodes and antinodes. So the Fourier transform of that pattern is essentially, if you do it, 3 dots, one DC component, and one a plus and minus the frequency of the pattern. So if we illuminate the sample, this guy here, with that pattern, then the emission function actually gets modified. So you actually start to see some of these moire fringes, or kind of patterning of the sample. So here you see actually some of the coarsest structures that we have seen on the example before. And interestingly, if you take the Fourier transform, you can see it's kind of a bit a mixture of information. So before, the Fourier transform has been kind of this radial spokes that came out of the origin. Now that in real space it has been multiplied by this sine pattern, you can see there are some spokes that have been displaced, and maybe if you want, so also this central dot has also been displaced. And this is simply because the multiplication becomes essentially a convolution in Fourier space. So you essentially convolve your sample spectrum with these 3 dots. And if you want, so you could imagine the convolution as painting something with a brush. And in this case, the brush has essentially kind of 3 tips. So if you paint something with that, you get kind of three copies of it. All right. But nevertheless, this is essentially the manifestation in frequency space of this mixing, of this down conversion of frequencies. However, when we observe them with a microscope, they still have to pass through what we call the optical transfer function, this circle here. We won't see the stuff out there. We essentially just see what's within that circle. But interestingly, if you look here at the top left, what goes through is essentially what normally goes through, plus 2 displaced copies. So information that actually belongs somewhere else. So you can see some of these spokes, they radiate out, whereas here in the DC component or the normal component, you can see with the microscope, you wouldn't get those. So what you then do in the SIM microscope in the post-processing step is you separate those information bands and you shift them to the position they belong to. So it's kind of like a puzzle, and you stitch that puzzle together. So in the end, from this one illumination pattern, you can essentially extend the coverage in reciprocal space. So you get additional information out to here. And if you measure it, if this is kind of normal distance to the cutoff frequency, oops, apologies, it can actually in most cases go out twice as far. Why can it go about twice as far? It essentially depends how fine that fringe pattern can be. So the finer the pattern here, the further out these dots are, and the further out these shifted copies are. However, that fringe pattern also obeys the diffraction limit. So usually these Fourier components or these frequencies also reside within the Abbe diffraction limit, or at best they can be at the border, and that causes essentially this doubling because the location from where they came sits here at the border, and then you add this additional information. You can also see, and I promise after this it will get less theoretical, you can also see that the extension in reciprocal space is essentially directional. It goes in the direction of the pattern vector. So if the pattern is from left to right, it extends in that direction. If you rotate the pattern by 90 degrees, you can obtain information in this direction. And then you can see you can probably stitch these together to get a bit more uniform support. And actually turns out if you use 3 pattern orientation rotated by plus or minus 60 degrees, then you can essentially extend almost uniformly the coverage out twofold. So this here is what you normally get through a microscope. And these are these additional information bands encoded by structural illumination and then stitched together to a pass band that is about twice as large. And this is actually experimental data from one of the seminal papers from Mats. So there were some directional features, like I think microtubules, and they had essentially Fourier components that radiate out. So similar to that computational sample shown here, but actually done with real data. And you can see that some of these side bands carry some of these Fourier components that radiate out from the center nicely, which otherwise would have been lost. So if you extend here, the larger you can extend here in frequency space, the number of components you can reconstruct or how far out you can be, the finer the resolution gets in real space. So if this is just shown here schematically, you get a sharper image, whereas if you only have the conventional information, the more narrow this gets here, the more blurry it gets. So typically SIM doubles the resolution because it can extend the transfer function out twofold. So in practice, this is how at least two-dimensional setup can look like. This is a combination of structural illumination with turf. So there's an evanescent field, and it is brought to interference to form a standing wave, so sinusoidal interference pattern, but also happens to decay in the third dimension. You can produce that by interfering 2 laser beams both above the critical angle. So if you were to look from the side, you would, well, this is very schematic, you would have this evanescent standing wave, and if you would look from top, it will pattern the cell just like this. And so what you need to do in a SIM microscope is you need to be able to phase step the pattern. This mathematical necessity. As I said, we want to try to separate the information bands. And so the number of information components you want to separate, that dictates how many images you have to acquire. Or without mathematical rationale, we can also see if there was just by bad chance a fluorophore in a minima, it will not get excited for this pattern position. So by moving the pattern, we can make sure that every fluorophore that is in the sample has been excited. Actually, if you do equally, if you, for example, do 3 phase steps with equal spacing, actually turns out that the sum of the illumination is uniform. And so you can make sure that you, from any fluorophore you have in the sample, you have actually obtained information. And besides the phase stepping, you also need to be able to rotate the pattern, say by plus minus 60 degrees, or here in the cartoon, by just 1 rotation by 90 degrees. And here are some examples of early turf sim imaging. These we acquired at the Janelia research campus. These are microtubules, here normal turf, and here with turf sim. And you can see that here again, this is about twice the resolving power to the normal turf microscope. And you can see that some junctures or overlap of microtubules get better resolved. And here, this was one of the first live sim paper from Peter Nair, also with Mats Gustafsson. Here, microtubules in a S2 cell, and here kinesin, here left in turf, and here in turf sim, imaged at video rate. And you can kind of see how these motor proteins crawl along microtubule tracks. All right, so hopefully this has been a helpful primer about light sheet microscopy in SIM, because now I want to talk why we were so interested in combining the two. So light sheet microscopy, as we said, it's very low in phototoxicity and photobleaching, and as such, it's very popular for long-term or rapid imaging. Like here, this beautiful work by Philip Keller, where they imaged embryogenesis in the Drosophila for, I think, several days. And the fluorescence doesn't go away. And actually, this embryo at the end can also hatch. So there is no signs of photo damage. So we love that about light sheet. However, the spatial resolution is rather modest. You usually cannot put the highest NA objective here. And it's also governed by wide field detection. So there is no additional mechanism to improve the resolution. And it's also a bit annoying that it usually uses quite specialized sample mounting. Like you have to put the sample in a cuvette or some other means to bring it in between the different objectives. And then the structural illumination, as we said, you illuminate the sample with sinusoidal interference pattern. You acquire images of different phases and different orientation. And when you then numerically reconstruct the SIM image, you can usually double the resolution here. This was work by Lothar Schermerle on chromatin organization in the nucleus, and you can see the much higher resolution, or also the definition here of the envelope, and they also looked at some of these chromatin structures inside. But what is interesting about SIM, or helpful, it's maybe one of the most biocompatible super resolution techniques because it doesn't use too much laser power. It's essentially a wide field microscope with the floodlight illumination, interference illumination. So the sample survives in wide field, it will likely survive in SIM. It has good spatial temporal resolution, so it can be used for live cell imaging or video rate imaging. It works with any dye or fluorophore, so it doesn't need anything special. So if it works on a wide field or a confocal, it will likely work in SIM too. But in its nature, if you image 3D sample, essentially illuminates everything. So what we said before, what we actually don't want for the photon budget, because we excite fluorophore above and below the focal plane. So we essentially waste those excitations. And if the sample is very densely labeled, this kind of floodlighting of the sample also leads to a lot of blur, which can then also lead to artifacts. So if you could combine the positives of the two techniques, then we could have a gentle 3D microscope that would also have a doubled resolution compared to what the light sheet can do. So why haven't we done that already? Because it's a bit tricky. So here, this is the business end of high resolution light sheet microscope, like a lattice light sheet. These are actually the objectives that are typically used. And somewhere here would the sample be on a tiny cover slip, like a 5 millimeter diameter cover slip. So this objective creates the light sheet, and here this NA 1.1 detects the fluorescence. And in principle, you can launch out of the excitation objective, the illumination objective, a structured light sheet, and that has been done in lattice. However, you can only have one orientation. So the pattern would run here from bottom to top, but you would not be able to rotate it. And as such, the resolution would be anisotropic. And there are also some limitations how fine that pattern can be, because the NA of that objective is not as high as this one. So you will also not be able to really double the resolution because the pattern is just a bit too coarse. So can be done, but has some caveats. So conceivably, we could also just add 2 more lattice light sheets from different angles and put the detection objective on top. So if we were to look from the bottom, we could launch a structured light sheet from here and take turns illuminating the sample. And if they're oriented by 120 degrees, we could essentially get those 3 directions that we have discussed before that would yield the kind of uniformly or isotropically extended OTF, optical transfer function, as such also kind of uniformly improved or isotropically improved spatial resolution. However, this would be quite a monstrous microscope, because you would have to build 3 lattice light sheet microscopes and weld them together and do that quite precisely. And there would also not be a space for the sample. So if the sample is on the cover slip, there is actually no orientation you can put it in that it wouldn't be in the way of some of the excitation or some of the detection light. So maybe you could put the sample on some sort of pedestal made of agarose or FEP film, but it would still be rather painful to somehow get it in the right place and making sure that nothing gets aberrated. So in short, could be done, but I wouldn't wish it on anyone to do it. Well, we would have been tempted to build one because it would truly be a very cool microscope, but I don't think it would be very practical, or at least with the engineering skills that I have. So for a long time, we thought, all right, would be nice to have, but probably won't happen. But then our lab got interested in a very different technique of light sheet microscopy, which are called the oblique plane microscope, which in short works like this. So imagine you have here a very high NA objective, say 1.35. So rather high angles can be produced by this one. And here at the cover slip, you launch out of the objective a light sheet at an angle, say 45 degrees. So the blue, the light sheet comes out of the objective and excites some fluorescence. Since that light sheet doesn't run parallel to the cover slip, it cannot be in focus everywhere if you were to place a camera somewhere here. Just some part would be in focus. However, more of it can be brought in focus by an optical trick that is called remote focusing. So whatever happens here is mapped, whatever happens here in this 3D space is mapped over here in, if you want, so a 3-dimensional image by this secondary objective. And if certain conditions are met, then every point along the light sheet can be mapped distortion-free and diffraction limited over here. So there is, if you want, so a replica of the light sheet formed here. And then you can build a tertiary microscope that just looks at that angle at this replica of what happens here. So if you do that right, you can actually form an image of the light sheet, even though it's at an angle that is sharp. And what is nice among many things is, yes, you can just put normal samples on here and enjoy benefits of light sheet, but it actually can also acquire the stacks 3D data very fast. Without going too much into details, to acquire a stack, you just rotate one galvanometric mirror, which moves the light sheet left and right, and it also descans the light that is returning, and so it puts always a sharp image on the camera, independent where you are. So it's similar like a confocal microscope that scans the laser spot, and the galvo mirrors redirect the fluorescence always into the pinhole. Here, just in 2D, it redirects the fluorescence onto the camera. Take it as a fact if you want, if my explanation wasn't good enough, which it probably wasn't. But here, for example, this volumetric imaging, this is a 10 micron scale bar at 13 hertz of a neuron labeled with GCAM. So you can really make this fly. If you were sample scanning, say in a lattice, I'm not sure if you can go over 10 hertz. You would surely start to wobble things. So we kind of started to like this, in particular also because there were some innovations by some colleagues at the Calico Institute. Calico was founded by the Google founders to study aging. And they also gave Andrew York and Alfred Millett-Sicking some money to build cool microscopes. And one thing they developed is an objective for this tertiary imaging arm, which is shown here, which again, without spending too much time on this, it has a bit of better collection efficiency. So you can imagine if you angle these optics here, then a lot of light coming from here from the left would just sail past it and wouldn't be picked up. But this objective has kind of glass frustum or snout at the front, which helps collect more of those precious photons and direct it to the camera. And as such, the resolution wasn't too bad, around 300 nanometers spatially. So similar, if you want, so to a lattice, but with the benefit that you have just one objective at the business end. All right, so this is OPM and also why we got interested in OPM. But what does that have to do with structural illumination? So what we realized is that instead of a single light sheet, we could also launch out of this high-NA objective 2 light sheets that would come out at an angle. This would look something like this. From the side, they would be propagating in the same tilted plane, but if you look at it from the back, they would essentially diverge from each other, an opening angle of maybe 60 degrees. That's roughly a numerical aperture of 0.8, so not too bad. And if they're coherent to each other, then at the very area where they overlap, they will form an interference pattern. So this simulation shown here is essentially what would happen if these 2 angled light sheets would interfere with each other. So you can essentially create a light sheet that is structured along its lateral direction. So this is a numerical simulation. If you were to look from the side, it would just look like a normal light sheet. If you would look from it from behind, you would see a fine interference pattern. So this was the realization that with this microscope geometry, the OPM, we could actually realize structural illumination, maybe even produce finer pattern that we can do with the lattice. And so this is how it would look like. So in the normal OPM, in the pupil, you have a line that is a bit displaced to the left. If that line were in the middle, there would be a light sheet coming straight out. If it's a bit displaced to one side, it creates a tilted light sheet, schematically shown here, as it comes out of the objective. And for the structural light sheet, we essentially use 2 line segments at the very edge or the periphery of the pupil. And they would essentially create 2 light sheets that propagate in the same plane, but are angled to each other. And when they're coherent, they would form this interference pattern. And then in the OPM geometry, we can scan them with the Galvo, and as such, acquire a volume, or more precisely, a parallelepiped volume of the sample under structural illumination. So this would, in capabilities, maybe be similar what you can do with the lattice. However, what you now can do is also rotate this excitation pattern in the pupil. So nothing prevents you from turning it by plus or minus 60 degrees. And that is something, as detailed before, would be rather difficult with a conventional light sheet because you would need to add more objectives. But here it turns out you can just rotate it in the pupil and as such acquire volumes under different orientations of the structural light sheet as required for a sim. And then you can stitch those volumes together, and where they have mutual overlap, you could do a reconstruction, a sim reconstruction that is kind of isotropic in all lateral directions. Okay, so this is how it looks as it comes out of the objective. So these 2 light sheets that interfere with each other, we rotate them by 60 and 120 degrees. And you could imagine it if you have a turf system with spinning illumination. So this is kind of a similar idea. You just spin them in the back of the pupil. And so also in real space, they change their azimuthal angle. There's a little caveat, because if this plane that they span or they propagate in rotates, then also the tertiary imaging arm would need to rotate, because whatever happens here is mapped over here in that remote space. And to form a sharp image, you always want to look orthogonally onto the light sheet plane. So in principle, we would need to build an apparatus that rotates this entire arm, which, yeah, I would also not want to do. This alignment needs to be rather precise, and once it's aligned, you wouldn't want to touch it. And it would probably also be prohibitively slow. So what we came up with is actually looking into optical elements that can rotate an image. One is this dovetail prism. And as you can see, if it rotates, it actually rotates the image by twice the rotation angle. and also has kind of bias rotation. So even if it's not rotated, it rotates the image by 180 degrees. So such an element you could actually buy from Thor Labs. You could put it into the microscope and rotate it, and you could rotate both the excitation and, if you want to, also rotate the image so that it always lines up as you would wish. However, this would still be slow, and a bunch of glass in a microscope can also cause dispersion and chromatic aberrations, so it would still not be that desirable. You can get the same effect with a so-called K-space mirror. This is an arrangement of, or K-mirror, my apologies, not K-space. So it gets its name from this peculiar arrangement of the mirrors and how the beam path goes. So the light bounces off this first mirror, goes to the mirror on the top, bounces back and propagates again straight. And as it happens, if you rotate that whole assembly along the z-axis, you also get rotation by 2 times the rotation angle. So if you rotate big gamma, the image would actually rotate by 2 gamma. So the idea was, very naively at first, proposed that we could essentially replace these 2 mirrors with scanning mirrors, galvo mirrors. And instead of having a mirror here that rotates, we could have 2 or 3 or more static mirrors at different rotation angles. And with the galvos, we would essentially select which mirror we would go. And as such, we could create discrete rotation angles that looked very straightforward on paper, but was also very wrong. So as you can see, the true K mirror will rotate along the z-axis, but these mirrors by necessity would rotate along a different axis. And as it turned out, it essentially diminished the overall image rotation angle. And with existing galvo mirrors, there was no way to rotate by plus or minus 60 degrees. So we spent some time during the pandemic thinking about other arrangements of these mirrors, because we figured out the math, or it's well known how you do these matrices that describe reflections. So you could think of other arrangements that maybe create, for a given rotation of the galvo mirror, a larger image rotation. So this was one of the harebrained ideas. And we also tested some in the lab, just to make sure that we do the math right. And eventually, we came up with a geometry that actually worked really well, and which is shown here. So in 3D space, there are these 2 galvo mirrors, and they're kind of angled in respect to where the light comes in by, say, an opening angle theta. And then if they rotate by alpha, the light will be deflected by 2 alpha, bounce off the mirror, come back, being descanned by the mirror. And if you then add different static mirrors at different positions, as schematically shown here, hopefully on the next slide it will become a bit clearer, you can actually impart quite a rotation onto the image, which is shown here, depending on the choice of theta and the scan angle. Typical galvo mirrors can do a mechanical angle of plus minus 10 degrees. You can actually find an arrangement that gives you exactly 60 degrees at plus or minus 10 degree scanning angle of the galvo. You can actually get more if you tweak the orientation of the mirrors, which is also an interesting feature. You can essentially amplify even the rotation much more than the K mirror or the dovetail. I see. I'm talking way too slow, I'm afraid. So hang on with me. My apologies. So let's just accelerate a little bit. So here, this is actually the practical image rotator. Let's just skip over there. There are some additional mirrors to make it practical, but in principle, there are the static mirrors and here are the galvo mirrors. So let's, here's the device in action, rotating this barcode. So we're looking straight into the image rotator. All right, let's just talk a little bit more how that then works in the microscope. So we insert the rotator in the OPM, here in the image space. So it rotates the structured light sheet and it derotates the fluorescence. So similar to the galvo, which scans the light sheet and descans the fluorescence, this device rotates and derotates. And so the camera would never notice that the light sheet has rotated or that it has been scanned. And here, this is some imaging of 50 nanometer beads, here left with the OPM, and on the right with the OP-SIM, which we call it, so it's a structured light sheet. And you can see numeric, if we put some values here on that bead, it more or less doubled the resolving power. From about 300 nanometer, it went down to slightly below 150. And here we also did a bit statistics over 100 beads, so it's fairly consistent over its field of view. And the Z resolution is a bit poorer, only around 500 nanometers. All right, let's maybe skip over that. I was just looking that it also works in a cell, and it gives you also with some decorrelation analysis a value around 150 nanometer. Let's just go a bit quicker. This was a sample that you can think of as a nano ruler that you can use. So this is a myosin motor complex with rod and motor domains that happen to form about 150 nanometer spaced structure. And it can just be resolved with the OP-SIM and also with an instant SIM, so structure illumination microscope, we can observe similar structures. So each time there is like here a rod flanked by two motors, that's essentially an event where we resolve one of these, if you want, so myosin nanorulers. Here, this is mitochondria. Let's again skip over the outer membrane, go a bit over it. So you could still argue, well, you can see it in the SIM, so why do you need the OP-SIM? So one aspect is that thanks to the light sheet nature, it doesn't photobleach as quickly. So this is 50 volumes with the OP-SIM, and here 50 volumes with the SORA, which is a spinning disk super resolution microscope with similar resolving power. And you can see that you know, after 50 time points, it almost entirely bleached out here, the clathrin-coated pits. The other aspect that we're excited about is that it can go really fast because there are no mechanical move or heavy parts, no scanning, sample scanning. It's essentially all galvanometer-based scanning, which can be fast. So this is kind of camera-limited volumetric imaging at about 1 Hertz volumetric rate of these mitochondria. And you can see these kind of nanotunnels or fine protrusions shooting out of some mitochondria, and you can resolve that without notable motion artifacts. And here, other fast dynamics are clathrin-mediated endocytosis. So again, this is volumetric imaging over the cell. And if we look here at the movie, this is also about 1 Hertz. And if you zoom in, you can actually see he's still a bit too slow. So some of the dynamics, when they really take off, they still get, you know, blurred out quite a bit. And these are the same region of interest in 3D. So you can see we can essentially also track or observe dynamics of clathrin-coated pits in 3D, whereas such movies before have been acquired in turf. So you essentially only saw the bottom of the cell. All right. I think I'm way behind. I don't know if people have to leave or go, but if people can still stick around, I can give a brief summary of the other technique I wanted to talk about. What do you think, Steve?
Yeah, it's all right.
Yeah. So my apologies. I was a bit sluggish. Or if you need to go, you know, maybe you can, if you're interested, you can watch it on the video. I will still try to be quick. So the second part is about a technique which we call the multi-angle projection imaging, which can be combined with various types of light sheet microscopes. And it goes at the heart of how we acquire 3D data. In a fluorescence microscope, we usually acquire a Z-stack. So a 3D stack of images, 2D images at different positions along the Z-axis. And then we plug the stack into graphics program and usually compute renderings, like different views, to understand it as a human being, because the stack itself is kind of meaningless for our brains. But here we can see that, you know, this cell has maybe in some direction some protrusions, and if you rotate around, we can kind of make some sense of it, of the 3D nature of it. So this is time consuming. It needs also a lot of resources. And if the data gets really large, it can also be difficult to interact. So we ask, can we essentially bypass the acquisition of the stack and directly get the projections under different angles? And if so, then we could be faster, we could acquire less data, it could be real time, or, you know, kind of video rate look at that as it comes out live. And if something were to happen really fast, where we couldn't acquire an entire stack, we could at least look at it in the projection. And this is how it works. We essentially rediscovered what is called the shear warp transform, which, if you take this image here, if you rotate it and project it, you get actually the same thing up to a scaling factor if you were to shear that image and project it. Let that sink in. Here we rotated it and projected it to the bottom, and here we sheared the same image. So two completely different operations. So here the nose gets rotated, here it just gets shifted to the left. But interestingly, this creates two projections, and if you put them on top of each other, they're actually to numerical precision the same. And here is some of the mass. Maybe I don't go full detail over it. This is the rotation matrix, and this is the shear matrix. And this 1, 0, 0, 0 here is the projection. So you project along this axis, which simplifies the matrix. And if you allow scaling, here this is a scaling matrix, projection, and shear, you find an expression like this. And if you choose Sx and Asx, these parameters correctly, you can find for any rotation angle a shear and a scaling factor that you get the same projection. Sounds very dry, I know, but it's still interesting that this holds true. So sure, if you rotate the image 90 degrees, you'd have to shear to infinity to get it. But say for rotation angle below 90 degrees, you will even practically always find a shear amount of shear that creates the same projection when you squish it. So why does this have any relevance to microscopy? Because we can essentially add that shear to a normal microscope, any 3D microscope. So normally, if we acquire Z stack, we stack the images along Z. And if you want, so we look at them in the Z direction. But we could also, while we acquire the Z stack, add an optical device that also shifts the images as they come in the X direction. So we would add a shear. So we can essentially, as shown here, shear the whole 3D stack and then sum it together. And we will see why you would want to do that. And first, how can we really fast shift the focal plane? Could use piezoelectric motors or voice coils that would get you somewhere into the 10s or 20 hertz. Or you can, like in the OPM, use this remote focusing, which in principle can get you into the kilohertz. And then you also need something to laterally shift images, which you can do with lenses and a galvo mirror, or what we actually prefer with two galvos. And if you rotate them together, you can also shift the image. And this is essentially what we put in front of the camera. So two galvos that can translate the image. And this is what you could do. So if you acquire a stack and sum the images along the stacking direction, you get one projection. But if you shift each image a bit to the side, you get actually a different view. That's within the shear warp transform. You can do that to any microscope stack you have acquired digitally. So you can try that. You can just shift in your computer program of choice the individual images of the stack progressively to the left and sum it and do different amounts, and they will give you different views. Or doing that in a stack is kind of silly, but what you can do is do it kind of analog. So here we move the focal plane through the cell and we speed that up quicker and quicker till we reach the frequency of the camera, which is about here. And once we're sweeping as fast as the camera integrates, we get essentially an analog projection. At once, we sum every slice together onto one camera frame. Looks something like this. This was really done just with a signal generator where we and by hand adjust the frequency till it was right. So here, we again sweep the focal plane, but we also add with the same signal generator a lateral shift on these galvas that create the lateral shift. And once we reach the frequency of the camera, we get an analog projection that is obviously different than this one here, which is more from the side, and this one here is more at an angle. And then if we change the voltages of the signal generator, so again, this is done without any computation, this is really purely analog. If we change the amount we shift images, we can actually create a rotated view with an additional stretching, with scaling. So if you go really to the extreme, the image also becomes a bit elongated, but nevertheless, you may appreciate that visually to our human brains, it looks like the image is rotating. And per the shear warp transform, it actually does. Okay. Here, this was one such analog projection in a single frame of cells. And here we acquired actually a stack and summed it together. And you may appreciate that the two views look quite similar. Again, this is some projection. So you can also, if you have the real stack, you can also do different projections like maximum intensity projections. So they would look different, but if you do numerically the same thing, which is done here in an analog fashion, they look almost the same. And here is the vasculature in the zebrafish, also done by this multi-angle projection technique. So every frame in that movie was a single camera exposure. And so we can essentially look around in the fish without acquiring a stack. These are just all single 2D images that are formed as analog projections of the sample. This can have multiple applications. So both in the lattice as also in the OPM, there is an annoying effect that you essentially scan your sample not along your detection axis. So your Z-axis is essentially not along the scan direction. And this causes this annoying effect that your data is skewed and you numerically have to de-skew it. And so if you were to look in a lattice light sheet or in OPM, and this is, for example, cultured neurons, it's kind of hard to interpret. So for once, every single plane is just a diagonal slice through your sample. And if you browse through it just through the slices, or if you were also to look at this data set, it looks just rubbish. And with the projection technique, we can essentially do the de-skewing and the projection at once. So as I move here through the sample, I immediately see a top-down projection. I could also change the angle. It could look kind of slightly sideways. And so this is directly in analog fashion, projected volume of these neurons, and one can immediately see that this neuron makes some connection or might be a happy neuron, whereas maybe this one was an unhealthy one because it has all these balls. And again, this was without acquiring any data. It was done live. Let's skip that for the sake of speeding it up. So you can also quickly acquire different views. For example, this beating zebrafish heart, the young zebrafish embryo, we imaged alternating at, say, 0 and 50 degrees viewing angle. So you get two views of this volume as the heart beats. This was done sequentially. You can also build an optical apparatus that can put on two cameras, two different projections. But nevertheless, if you were to acquire a stack, you would have a hard time keeping up. So if you would acquire hundreds of slices, they would not be synchronous. And so your view of this 3D sample would be kind of blurry or jagged. Here, these are some other fast dynamics in the Drosophila larvae. So this guy has hatched. This is a myosin labeling. I think these are some muscle cells contracting. It's still just a subset of the whole larvae, but again, this was imaged at around 10 hertz. So also here, for the very fast dynamics, you would simply have no chance to acquire an entire stack to even get those views. Whereas here, each frame was a single camera exposure. And again, this covers a certain depth, a certain volume that was acquired here with the OPM. And even faster, this is viewing a cultured neuron at a frame rate of over 100 hertz. So one can see the calcium dynamics by also changing the viewing angle. All right, and so this is kind of cool. We're still working out what we can do with it. So we're also thinking about reconstructing 3D data from few numbers of views. So for example, pinpoint where neurons are in, say, a Drosophila or zebrafish brain. But we can also use it for the OP sim. And this might be, let's just try to do it here. So before in the OP sim, we had to acquire volumes under structural illumination from different directions. But with the projection technique, we could, in an analog way, create projections that are exactly along the z-axis, so the axis of the detection objective or parallel to the cover slip of these 3 volumes under different rotations of the structural light sheet. And hence, we could essentially just do the sim reconstruction, also the fusion of the data, just purely in a 2D plane, whereas before we had to do it in 3D, and so using hundreds of images, here we could do it with much less. So this is done here. These are actually projections of a cell with mitochondria under the 3 different orientation of the oblique structural light sheet and squished or projected exactly into the same coordinate frames parallel to the cover slip. So this is how the raw data looks. But if we co-register them, so squish them, rotate them, overlap them, then this actually looks much more like traditional sim data. If you could zoom in, you could actually see that the patterns run in these different directions, and when you overlay them, the data overlaps quite nicely. And so here, this is a reconstruction from the projection, and this is a 3D reconstruction from the OP sim of these mitochondria structures. And if we zoom in, here, this is the sim reconstruction from the projected view, and here from the OP sim. So these structures are different in heights by maybe 6 microns, and you can see that they're kind of captured, to some good similarity. Sure, maybe some structural differences from the projection come here different out from the color coding, but overall the structures are quite similar. So with that, you can then do projective sim imaging of the entire volume. So again, this is different from, say, turf sim that just captures the bottom. This captures the entirety of the cell, squished the whole volume into 2D, and then sim reconstructed. So mitochondria that are at the bottom, or vesicles, and also mitochondria on top of the nucleus come into view at once. All right, if you're still on this call, then I appreciate your patience. I leave the discussion away and go straight to the acknowledgement. And again, thank you so much for sticking around, and hopefully I can still answer some of your questions.
Thank you, Reto, for the extensive coverage over the different modalities. I have a couple of questions, but I'll leave it for who's on the call to ask some questions first.
Sure, maybe
if anyone's
yeah, again, my apologies. Somehow I used to be able to give that talk in like 20 or 30 minutes and thought, yeah, just a little theory slides, but then I guess I got carried away trying to.
I like the bank. The bank is pretty good. Yeah.
So hopefully there was something useful either for the sim or for the fluorescence bank.
So I guess anyone has questions, you could just unmute and ask. Yeah.
I had a question. So for the when you were talking about doing the interference with the 2 sheets and rotating them around, you generating? So my first question is, how are you generating those sheets to generate those lines in the focal plane and to switch through? Are you using an SLM in that case?
Yes, I left that away, so my apologies. At the moment, this might be difficult to explain without the appropriate cartoon. So in short, no. We use currently a Michelson interferometer with which we can create 2 beams, and each beam can be controlled with the mirrors in the interferometer to be positioned where we want it in the pupil. So this is quite hand wavy, I'm sorry, but essentially think of a beam splitter that creates two beams, and each beam is individually controlled.
Yep.
Okay.
Maybe I can find the drawing, but yes. But if you would want to make more complicated patterns, then yes, an SLM would be preferable. So one could think one could maybe even try to produce some type of optical lattices. So you could, in a similar way, like the lattice light sheet, use an SLM that shapes kind of the illumination beams, and you somehow have to offset them. So in the lattice would have its order centered, so you have to shift them to the side. But that you can, in principle, do by either having some mirrors or others, because once they are at the edge or wherever you want them, and the image rotator is correctly oriented, then it will just rotate them. So that's kind of the beauty. If you have them somewhere where you want them and everything is correctly aligned, it it just rotates them. If it's not correctly aligned, maybe one of your orders will jump over the edge and disappear.
Yeah. So, and I guess like the alignment, with the alignment as well, like what you're talking about here. So, I mean, I know even with like a 2D sim or a 3D sim, the alignment can be quite, you have to get the quite precisely right.
Yes, that is correct. If you have a turf sim and, all of a sudden one order is clipped or falls into epi, yes, that is correct. What shall I say? It makes it not easier, that's for sure. But we figured out some sort of alignment protocol. So we treat the image rotator as a separate module that you can take out and put it on a separate optical table and place some pinholes at a meter away on each side and just observe or align that a beam that goes straight in doesn't deviate by much over, say, a meter of propagation. And so that way we dial that thing in that it has a very small angular error. And then it works, at least in our hands. So the beams don't disappear or in one direction all of a sudden change the tilt. So if they would move in this direction a bit inward, then the plane would be differently tilted, and we can observe with some test samples that the waist of the light sheet stays kind of where it should be and doesn't tilt relative to the tertiary. So I think it's manageable. If you will build a turf microscope, I think it will probably, you could build a turf sim microscope with that rotator if you would want so.
Cool.
And I think the turf will be more sensitive. If you have a little angular error, you will probably see it because the penetration depths would jump around. So maybe one could do it, but I think it will probably push it even more in this application.
Thanks.
That makes sense.
Yeah, makes sense. Yeah, thank you.
Tina, do you have anyone any more?
Yeah, just on top of the same concept, I'm wondering if the fringe pattern is actually scanned across the field of view.
How or if?
Maybe I missed this part in the talk, but I'm not sure if the interface